Hello everyone, welcome back to Alkaline Hydroxide 784, and today we are once again in the GP overview series. I had like 4 suggestions and they were all in science, so for now we'll just go with science. Okay, so today's topic is gene editing. Maybe next week I will probably do a different bigger categories, like a different one, maybe like arts or something, and then just come up with a topic that I think might be worthwhile explaining. I don't know. But yeah, anyway, today's topic is gene editing. So hopefully this will be shorter than the first episode. And yeah, you can also comment on whether you prefer the first episode or this one. Okay, let's start. So, for gene editing, the first thing I'm gonna talk about is what exactly it means, or the different kinds of gene editing. So, to me, I am not gonna really refer to a specific website or anything for what what is actually counted as gene editing, but to me, gene editing is basically changing the DNA sequence in some way or the other. It can be like directly swapping out some of the base pairs for a different bunch of base pairs, or it could be adding some extra DNA to the gene. So therefore, I will be using various kinds of I'll be talking about various kinds of gene editing throughout history, and it's not just CRISPR that we're going to talk about. Okay, so the most oldest form of, like oldest way of modifying a genome or like genetic modification is actually selective breeding. So, in fact, a lot of, your everyday dog, like your household pets, they were selectively bred. Like for dogs, especially the earliest ones are actually wolves. There wasn't something called a dog until, or maybe there was, I'm not sure, but the household dogs come from wolves long 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 ago, who were actually quite friendly to the humans, and humans liked them, so they kept them and they bred them, and yeah, now we have so many varieties of dogs today. And also for plants, like selective breeding is used to make plants more nutritious or bigger or something like that. So like they choose the one that, for example, if they like this particular apple tree that makes really nice tasting apples, then they'll make sure to keep the seeds of one of the apples, and then they'll grow trees of those specific apples in greater quantities compared to the trees that they aren't very into. Then they'll probably throw those seeds away, or they'll try to not let them become trees. I don't know. Yeah, that's how selective breeding kind of works. We'll be talking about them more later on. So the next thing that came, which is a bit more recent but not super recent, I mean it is there within our lifetime, maybe my lifetime, or even, yeah, it's probably around that time, very early 2001, it was already there, that kind of thing. So GMOs, like GMOs were there for quite a while. I think even before 2000, I'm not sure. I didn't bother looking at the history of when exactly they started. But GMOs are genetically modified organisms, and that's what people were talking about before CRISPR came. So, you might as well give it a mention. So GMO, it usually comes up in the topic of, do you buy GMO products or not, right? So, but what exactly is a GMO? Well, a GMO is when you modify the gene of some particular organism, usually a plant. But sometimes they do stuff to salmon or something to make them grow faster. But yeah, usually it's plants, and they do it by using a gene from another organism. Most of the time, it's actually from an animal. So they implant that gene into the plant gene in order to give the plant certain properties that it otherwise wouldn't have. And the idea is that these properties would make it more resistant, or like make it better in some way that helps, which is more beneficial for the humans, like the people who want to eat the plant or the whatever that is. Yeah. So we'll look at that as well later on. So, how it's usually done is using a bacteria. So the bacteria has a gene inside it. So usually that may be animal genes, or it may be bacteria genes as well. So the bacteria has a gene itself, and they put it into the They make it do something to the plant cells and it gets transferred into the plant cells. Or they have this gun thing that shoots gold nanoparticles into the cell and that transplants the DNA in there as well. Yeah, that's pretty crazy, but yeah, okay. And the latest stuff we have today is CRISPR. So CRISPR is the thing that people are talking about nowadays in the news, in everywhere. Okay, maybe not right now, but like 2 or 3 years ago, they were definitely talking about it. So what exactly is CRISPR? So CRISPR is actually kind of somewhat influenced by what we do in GMOs, but the difference here is we use just the enzyme. So we have the enzyme that comes from the bacteria, and the enzyme helps to cut out a certain part of the DNA, like the DNA strand. So it will find, maybe it wants to find CCGET or something. Once it finds that, it will go and splice it at that point, which means it'll cut it in a way. Either it will produce a sticky end in which one of the DNA strand is sticking out and the other one is not sticking out, so one of them is slightly longer, or it will cut it nice and neat where both of them are exactly the same length. And after that, they'll bring in the new gene and that will be attached using DNA polymerases, which I think you know what they do. So they just stick the stuff back together and boom, you have a edited DNA. Yeah. I think it would be better for you to just read or watch the whatever links that I put below, because that would give you a better understanding of CRISPR. Even though I am studying bioengineering, I feel like it's better for you to just look at that information on your own, because it's not very relevant for GP itself. The thing that's relevant for GP is the issues that this could cause. Yeah. So for CRISPR specifically, the enzyme people mostly talk about is Cas9. But there's actually other such enzymes. So there's like Cas11, Cas12, and there's some CPR1 or something. Yeah, there's a bunch of different enzymes that can be used. And here's another method that I'm not sure how recent or how old it is, but it is something that we are quite familiar with. If you do enough biolabs, you will actually encounter these ones. So this method is called, I'm not sure whether you should call this the method or the product, but yeah, it's recombinant DNA. So what this thing is like, these, there's this thing called a plasmid. It's like a DNA that's in a loop. Like it's not that it doesn't have two ends, it just connected to itself like a loop. And these plasmids are usually found in viruses or bacteria or something. So basically what the biologists or the lab people do, they take these plasmids, then they cut it at one spot, and they can add some other DNA in between, inside that spot, and lyse it together again. Sorry, not lyse it, glue it back together, which is kind of like CRISPR, right? But it's a bit different. It's a bit different, because the CRISPR, they usually refer to the normal DNA kind of strand. This one is actually a plasmid kind of thing. So this is called recombinant DNA, and you're actually adding on stuff. You're not removing anything most of the time. You're just adding on stuff. And you have to add it to specific locations based on where the origin of replication is and stuff like that. So the origin of replication is the point where the part of the DNA where the the stuff that makes new DNA, like the polymerases, it will find that location and then start making DNA after about 50 nucleotide pairs or something like that. Yeah. As I said, I think it's better for you to just read this, because if I explain this in greater detail, I'll take forever. So yeah, okay, cool. So plasmids are quite useful for medical purposes. So usually the most common example I can think of is insulin. They can incubate bacteria to make insulin. So all these bacteria, they put the plasmid into one bacteria, then just keep growing it into many, many more bacteria, because bacteria reproduce quite quickly. So then you can just incubate them in a big thing, like that, like a big container, and then they will produce insulin. Yeah. So it's a bit easier to use that compared to the actual location of the insulin. So like for example, they take the insulin genome from a pig, because, or it could even be a human one, because actually trying to grow pigs to create insulin, it seems a bit wasteful, so they just use the bacteria instead, which is way better, honestly. Yeah. Yeah, then they can just use it for medical purpose. Right, so this is, it can also be used for antibiotics and stuff. So you can you can look that up on your own if you want to. Yeah. Okay, so fine. There's a lot to know when it comes to gene modification, editing, or whatever you want to call it. But what are the issues surrounding these techniques? Like, what is the stuff that we actually care about? Okay, to me, I would probably identify three things. The first thing is practicality, second is ethics, and the third is accessibility. So first you'll go to practicality. Practicality means like the functionality of the methods, like what you can use it for and things like that, without really looking at other aspects, just very basically what it can be used for. So CRISPR is quite new, I guess, at least compared to selective breeding is definitely new. It has a lot of potential. There's actually more and more studies done on CRISPR as time progresses, or at least in the last 2-3 years, there's been quite a few studies done on CRISPR. And but at the moment, it's not super useful yet, because it's a bit there's a lot of concerns regarding CRISPR. We need to see whether it's safe to implement and stuff like that. So we can't just use it at the moment. We have to wait for some time before we can use it. So it has a lot of potential, but not very useful yet. At the moment, the only thing I think CRISPR is really being used for is gene therapy. And yeah, so in gene therapy, it's basically done in adults, and CRISPR can be used to edit some of their cells to have a certain effect, so it can cure certain cancers and things like that, to help the to help with to just help cure the cancer, I guess, to help the immune system fight the cancer, I guess. I think. Unfortunately, I'm not that familiar with it. Oops. I only know how it's done. I don't really know how it works in a cancer setting. But you get the idea. So yeah, in that scenario, it is actually okay. If you when we move on to ethics, I won't really talk about this in ethics, but technically this one is okay to do, because it doesn't impact any other person, or it doesn't impact all of the cells in that person. Yeah, it only impacts that local, it's like very local kind of therapy in a sense. So that's what CRISPR can be used for at the moment. But in terms of theoretical use, like theoretical use is to remove genetic diseases in an embryo, like before it even develops, becomes a baby in a sense. So that's the technical potential, the theoretical potential of CRISPR. And of course, it has been demonstrated, as we will see later, but for the most part, it is not being used for that at the moment. It's only being used for gene therapy. Okay, so the next thing that we can look at is the other methods. So what are these methods actually used for? Just general gene modification. The first one is food optimization. As I told you earlier, you can actually do many things with plants, as you can see, GMOs, or even just selective breeding. The reason why plants are actually quite featured is because they can handle polyploidal DNA very well. So basically, if a human being suddenly got an extra set of chromosomes from their mom or their dad, it would be quite disastrous. But in a plant, it doesn't matter. It still grows. That's why you can actually just insert random DNA into the plant and it will still grow and not die. But in an animal, it will be quite disastrous because it may not work most of the time. Especially if the DNA is like a foreign DNA, is definitely going to create some problems. That's why you had to be very careful with actually doing it, because most of the times it doesn't really work. And usually the selective breeding is used compared to doing GMO or CRISPR on animals, because that's just definitely easier to do. And yeah, it's done within the same population, so you don't really worry about foreign DNA as much. So in plants, yeah, we use it to make corn from tiny little thing in the past to whatever size that you remember corn being today. And even watermelons and other fruits as well, like apples are made sweeter and other. I think that's all I remember, sorry. But yeah, you can find a lot of examples like this online. And wheat as well is actually a hybrid grass. So it's actually a hybrid of two different grasses, and it is definitely not the normal amount of chromosomes that a plant would have. It has more than that. So as you can see, the plants actually still survive despite having so much of DNA in it. Yeah. Um okay, so something that we can something that we look at is eugenics. It's actually the same as it's another way to describe selective breeding, is to selectively select certain traits in a population. So we will look at the ethics of this later on as well. Yeah, so as I mentioned below, before, it helps with making better plants, and also with with the dog thing. Like you made so many different breeds of dogs, or at least selectively bred them in a sense. Okay, so apart from those kinds of stuff, another practicality that we can get here is health. So I did mention plasmids earlier, right? So plasmids actually help us a lot with health related stuff. So you can make biologics out of them, and yeah, or you can, basically by biologic I mean like biological medicines in a sense. Yeah, I'll tell you more about that in a second, but it's useful for that kind of stuff like insulin, antibiotics, etc. Yeah, I think that's pretty much it for practicality. There are probably other things that it can be used for. If I do find them, I will put the links in the description, or I mean in the website that I have. Yeah. Right. Okay. So the second part is ethics. Um, so the main questions in terms of ethics is usually meant for CRISPR. So I'm gonna start with that actually. So the thing that people ask about CRISPR is whether the whole designer baby thing, is that gonna be a problem? Well, technically we already have the fact that people can select the best embryos when they're doing IVF, that procedure. Yeah, they can actually ask the doctors to check if the embryos are likely to develop a genetic disease, and then just choose the one that doesn't have it. Well, do you think that is ethical? I don't know. It could be unethical, or it could be done in good sense to prevent the kid from having a disease. Like, choose the one that doesn't have it. But really, that's already an option, even without using CRISPR. It's already like that kind of selection is already possible in patients, in a sense. If you have enough money, that's kind of possible. But for CRISPR specifically, like to make it even more controlled, like even more specific DNA of the kid, well, yeah, there's a lot of problems with it, because the first thing is that we do not know whether it's safe, as I mentioned earlier. That's why CRISPR isn't that well used at the moment. So that's the first problem. The second problem is whether if you did manage to edit the genes and the kid grows up and then they decide to reproduce, would the edited gene get passed down? What happens next? Does it lead to engineered evolution? Technically, we are already engineering evolution by selective breeding, but in this case it might be more drastic, because it's like deliberately putting in certain genes and directly from the get-go. We don't know what that would lead to, whether that will cause certain defects in the subsequent generation, or whether that may cause other problems. That's the second problem. The third problem is in terms of societal disparity. So if in of course many this is more sci-fi kind of scenario, so if you imagine in the future designer babies were definitely available in the market, then who would actually be using that? Well, the rich people, of course, not the middle class, not the poor. It's usually just the rich people who would be using this technology to their advantage, and that could lead to people who are basically like, in Olympics you have rules against using steroids or stuff like that, right? But in this scenario, you have people who are already having the genetic advantage even without taking steroids or anything. Is that unfair to the other players, or what does that mean? So there's actually a lot of things you can ask based on this ethical thing. And yeah, I didn't take any source for this. I've seen this information before, like I've seen that kind of question before. If I do find the video, I'll put it. I'm sorry. Okay. But yeah, those are the things that you would need to think about, because if let's say the parent can edit the gene such that the kid is very athletic, very smart, very everything, good in everything, then that kid will end up being like the top 1% in the society, and that would lead to even more inequality. Like we already didn't have inequality today. Right. Okay. So yeah, that's the idea. That's the problematic ethics behind CRISPR. So in real life, the thing that we've seen is this China doctor who did this experiment to see if it was possible to edit the genome such that the kids have a lower incidence of HIV or something. And he actually did that in two. Oh man, okay, never mind. Just ignore the background noise. He actually did that successfully in a twin girls in a sense, and we do not know what will happen to them or whether they'll be fine or how things are gonna go. But he did it, and because of that, he got jail term. So, I would suggest you don't do that, at least for that reason. So, in other words, I'm pretty sure the law doesn't allow CRISPR to be just anyhow used. You need to do proper research before we move forward with this technology in a sense. Yeah, but anyway, I feel like it's actually kind of a dumb reason to edit genes, because you can avoid HIV in many other ways that do not involve CRISPR, and probably not that hard to do those methods anyway, so I don't get why you would want to edit a genome just to avoid HIV. It's pretty stupid. Okay, anyways, let's move on. So if we ignore CRISPR for now and we talk about other already existing methods that have been in use for a long time, we will now come back to eugenics, or selective breeding as you would call them. Of course, it seems like it's a good thing. It gives us nice plants. It gives us many different kinds of dogs that we can adopt as a pet in a sense. But there is some problems to eugenics itself, and that usually occurs when you apply it to animals. As I mentioned earlier, if you do it in animals, it usually becomes a problem. But if it does succeed still, the results can be quite scary or terrifying in the sense. For example, there's this kind of bull called the double-muscled bull. It is an abomination in my opinion, and I feel it's not very ethical, because basically what they do is they make the bulls have twice the amount of muscle that a normal bull will have, so that they can get more meat from it. But because of this, when they are born, you literally have to do a C-section on their mom, because they're too big to come out. And that is already very weird. Like, that just sounds unnatural already. And when they grow, you can literally see how unnatural it is. It just feels uncomfortable, to say the least, in a sense. But another example, which is maybe less scary but still pretty sad, is actually the bulldog. Those cute little dogs with the flat faces. I mean, they look kind of cute, but the problem is that their face prevents them from breathing very well. Like their nose is literally squished into their face, so it's hard for them to breathe. And as much as it is nice for humans to, they like the look of the bulldog, it's also not nice for the dog itself. It can't breathe properly like not like other breeds of dogs. You know what I mean? Yeah. So in a way, is it really ethical to be doing this? Then, yeah, I'm not sure. It's probably not that ethical to do it on animals. Then what about plants? Why do I not care so much when it comes to plants? Because I guess maybe because there's plants. And also plants don't seem to really care if you put a bunch of DNA in them anyway. And we have done way too much anyway. Like we've already done so much when it comes to plants. Like if you've seen what old corns and watermelons look like, you would know for sure. We have done a lot. And it doesn't seem to affect us in any way either, because humans have been eating these selectively bred plants for ages, and therefore there's a possible argument to say that maybe GMOs aren't as terrible as they seem, because there are definitely people who raise a lot of who weren't very into GMOs when they came about. In that sense, maybe the difference between the two is that the selective breeding uses the genes from the same population or from similar populations of plants. But when it comes to GMO, they usually use DNA from bacteria or even animals to implant into the plant DNA, which might lead to consequences in the plant. Or will it actually lead to consequences? I'm not sure. So yeah, but for now there aren't any big issues because there's a lot of you know regulation that prevent something terribly dangerous from happening. But maybe in the long term there could be some problems with GMOs. But the thing is, GMOs have been used for very good purposes. Like for example, golden rice is actually a GMO, and it has a lot of beta carotene, and that's pretty useful for those in places like Africa. Oh great, it's gonna be 30 minutes again. Great. Okay, never mind, sorry. It's useful for in places like Africa where you can't really get a lot of food, and this beta carotene will help give an extra vitamin boost in the rice itself. And that's pretty cool. So yeah, there's definitely many examples of GMOs, and most of them are done for a beneficial purpose. Like the intention is to create a beneficial result. They don't want to purposely create a carcinogen or to create something that can be harmful to people. They want to create stuff that's useful for the world. So in a way they aren't really being unethical, but it's the bigger issue is whether the GMO themselves is safe at the end of the day. Yeah. Anyways, so the third aspect is accessibility, which is quite important actually, because if you can make it, that's cool. You can use it for many things at school, but if people can't access it, then what's the point? Yes. So for now, for this segment, instead of talking about gene editing itself, I'm gonna look at a more broader topic of biologics, because it's easier for me to just talk about it that way. And this would include stuff that's not necessarily related to gene editing. Yeah. So what is a biologic? Well, if you've heard of pharma, you've heard of the increasing prices in pharma, allow me to introduce to you the biologics industry. It's like the bio version of pharma, but instead of synthesizing stuff with traditional chemistry, if you would call it, we are doing stuff using cells, bacteria, or viruses for that matter, using biochemistry instead of just chemistry. You know what I mean? Yeah, so technically it's still chemistry, but it's biology more of. Right, so if you've ever done a biolab, you would know what I'm talking about in terms of biochemistry, and you've learned firsthand that this kind of stuff is actually quite hard to do. Like for example, growing bacteria, doing a simulated PCR, or doing literally anything with a DNA is really hard, because even the smallest mistake you do is gonna create a mess, like gonna make it not work. So it's even more problematic compared to purely chemical I mean, not purely chemical, as in more traditional chemistry stuff. And therefore, you need a lot of care to get something viable. This means that it's actually quite expensive to make biologics to begin with. And that's a problem here. We have increased price in biologics, and that means people can get it. But the other reason why both pharma and biologics are expensive, which I think is quite interesting to know, is because generally the company has a limited patent time, about 20 years. And the funny thing is they have to take the patent from even before they start clinical trials, so that other people don't steal their idea in that time that they do all of the clinical trials. But by the time they finish the clinical trials, they have wasted at least 10 years doing all of the trials. And I think it's not really because of the length of the trials, but because of probably more of regulations and other problems that force them to use the patent at the beginning itself. So by the time it gets to market, they already wasted half their patent time. So they want to make the most of the rest of the time, and they will tend to charge more for the product at the beginning. As long as they have the patent, they can charge as much as they want. So they will charge it as much as they want for the 10 years until it becomes patentless and becomes a generic drug, which will become way more cheaper and become produced in like some other countries. Like other countries that okay, never mind. Yeah, another country. So for pharma, that's already a bit of a mess. But for biologics, it's even worse because, cells, yeah, that makes it even harder to do things and it can be even more expensive. Like, so the thing about cells is that clinical trials will be even more stricter and the requirements will be even more stricter in a sense. And that would require even more time to go through everything and actually put it into market. So the next point in accessibility is the location. In terms of doing biologics, you actually need certain lab equipment to do biologics. If you've done a bio lab, you know what I mean. You have some fancy stuff to heat the samples, to rotate it and do all the stuff in the bio lab. And some countries would have a greater ability to do those things, especially the stuff that has the patent. So usually the big companies would be having the monopoly over the stuff that still has patent. But once it becomes a generic stuff, like once it becomes a generic medicine, oh well, you're gonna be seeing it in India and China, mass production there. But actually, it's not a bad thing, because I think in India and China, since they can mass produce not just for biologic but also for pharma, they can produce a lot of medicines at a much cheaper price, and it's still pretty effective. It's still the same thing. It's not like it's worse in quality or anything. It's still the same thing and it's much cheaper, and that's pretty much a big contribution to the actual industry, and it helps a lot of people access it. So that's probably a good thing, huh? Right, okay. I mean, in the point of view of the public, for the company, it's probably not a good thing because they don't get money from this. They only get money from the stuff that they themselves sell. Yeah. Okay. So the last part of accessibility is, of course, politics. Because even if you can make stuff and do stuff, you need someone to allow you to do the stuff. So, yeah, like the first thing I've written here is like a fake laughter in a sense. So as you can see, even in terms of when I was talking about the price earlier, regulations do control a lot of things. They force the company to have a patent from the beginning itself and do all that stuff. Right? Of course, clinical trials are important, but I feel like some of the regulations tend to prevent them from maximizing what they found, like whatever they can get out of their idea and their patent in a sense. And they are forced to end up overpricing the product before their patent ends. But anyways, for actual politics, I saw something called this USMCA deal, which was a replacement of the free trade NAFTA agreement. And yeah, apparently it affects biologics in a way. So you can read the article on that because I don't want to waste time trying to explain it because I don't understand it myself. But anyways, it makes it harder for the biologics industry to do stuff. And as another final observation that we can make in the current situation, the virus in the sense, we see that the retraction of certain regulations allows the research on the virus itself or on vaccines or cures to be faster, and more stuff is getting done, like more stuff is being done in a sense. Yeah, so I'm not very familiar with what the exact regulations are, but I do know that some reform might be needed to get things faster, like things to happen faster. Clinical trials can be processed faster and stuff like that. But of course, that we must also know that as much as they seem to be really strict and all that, they also have loopholes that let dangerous products through into the market, and that definitely happens. So in a way, regulations are both overly strict and yet not sufficient. Oh well. So I think in the future they should probably be made such that they ensure safer products, but at the same time allow clinical trials and stuff to happen faster so that people can more efficiently access the safety of whatever they're making and do things quickly. Yeah, so here's the conclusion. We'll hear the conclusion, and I hope this gets your brain juices flowing, or at least figure out what other stuff you should search or Google about when you want to know about this topic. Because honestly, I'm only familiar with the sciencey part rather than the politics. Yeah, I have nothing to say about that. Oops. Okay. Yeah, that's about it. You can see the links in the website. Unfortunately, I couldn't make this shorter than 30 minutes. In fact, it's longer than the first video. Oh well. Never mind. I hope you find this video useful anyway. If you did find this video useful, you can press the like button and subscribe. And with that, I would like to say thank you and bye!